When I asked my toddlers what they wanted for breakfast, they said they were craving toast, so I woke up at 4 a.m. to get started on the bread. I realized that I didn't have yeast for the recipe, so I had Lucky go out to forage for S. cerevisiae yeast for me.
I started by spraying the surface of my gloves with ethanol to avoid contamination, while Nara unwrapped a fresh, sterile yet plate and labeled it with the grid of the mutant strains and started gathering toothpicks for the cross.
He came back with 4 mutant strains that all had a red phenotype. The yeast can have the same color, but that could be due to the mutations in different genes. My kids refused to eat any bread made from yeast that have mutations in different genes, so I had to perform a complementation test from scratch to find out the genotypes. No, Lucky, I need a fresh, sterile toothpick. I tapped on the HB2 colony to get a small amount of yeast cells and started streaking for the parental patch. I then went down the column and put a spot in each row for the crosses. 1 toothpick full should be enough, and to know you did it right, you should see marks left by the toothpick, but not clumps of cells.
This recipe was passed down by my grandmother, and the key advice she always gave me is more is often better, but not in this case. If there are too many cells, instead of a homogeneous patch, there will be sections of diploid and haploid.
After that, I got an HA2 strain, streaked the parental, and using the same toothpick, I mixed it with an HB1 spot to perform the cross. Flipping the toothpick over, I streaked the patch out. I then repeated this process for the remaining crosses.
Yeast are motile, so they need to be mixed in patches so they can mate.
Excuse Lucky, he likes to talk about mating. When 2 compatible yeast mating types are close, they release pheromones. Alpha cells secrete alpha pheromones, which causes nearby cells to grow towards them. This leads to the fusion of their plasma membranes and nuclei, completing the mating process in about 4 hours, and that's how yeast mate. I then incubated the plate at 30 degrees Celsius with the lid down.
What are we supposed to see from this?
If the mutations are in the same gene, the diploid yeast formed from the cross will not inherit a functional copy of each gene, and we'll see the mutant red phenotype. This happens because the genes fail to complement each other.
So if they're in different genes, the diploid yeast will have 1 wild type and 1 mutant copy at each gene, and will thus show the wild type white phenotype?
Yes, because the genes complement each other and supply the functional missing product. When I came back, there were white phenotypes, which meant they complemented. There were also mutant red phenotypes, which meant they failed to complement. Therefore, I went ahead and used the red crosses for my kids' morning sourdough.